Hello, everyone. I'm really delighted and honored to receive the Andrei Borisovich Vistelius Research Award. I'm heartbroken I cannot make it there in person to receive this award, but believe me, I tried everything I could to make this trip happen. Let me start by thanking IAMG and the awards committee for this incredible honor. It does feel wonderful to be one of the IMG Early Career Awardees when there have been so many winners who've gone on to have such illustrious careers. I would also be remiss if I didn't call out at least a few people for this award. I'd first like to thank Peter Fox, my PhD advisor, who took a chance on a young master's student with very little confidence in himself or on his big ideas. To my wonderful colleagues who nominated me for this award, thank you. I don't exactly know who all nominated me, but I do know Bob Hazen led the nomination. So thank you, Bob, for this nomination. And really, thank you for shaping my career through the past 9 years of collaboration. Bob is one of the many colleagues and collaborators from whom I learn every single day. As a data scientist who has set foot into one too many fields, I'd say, I thank every one of my colleagues who welcomed me with open arms into their area of research, who patiently taught me about the cool things in science. So thank you to Shauna Morrison for teaching me about mineralogy, to Mike Wong to teaching me about astrobiology, to Yulia Hubner to teaching me about material science. Really, the list goes on and on. I'd like to thank all of my collaborators who make my work a joy every single day. I'd also like to thank my group at Carnegie, who makes my work so fun and impactful. You guys sure keep me on my toes, but keep telling me about the new ideas, projects, and solutions you're developing. Your enthusiasm for science is infectious and certainly improves my day during the tough times. There are so many others that I'd like to thank. I might as well keep going for the whole 60 minutes. But it's science you all are here for, and it's science I shall deliver. So, to reintroduce myself, my name is Anirudh Prabhu. I'm a research scientist and PI at Carnegie Science. I'm a data scientist by training, and for the past decade or so, I've been developing algorithms, models, and methods to answer some of the biggest questions in science. My work has mainly been applied to the fields of Earth, space, and life sciences, and everything in between. So I want to start by showing you all the projects that I have actively going on right now. I'm leading a bunch of these projects and I'm collaborating on others. Well, here they are. As you can see, it's a pretty busy time in my career, so I'm pretty happy and excited to be working on all these projects. Today, we'll be focusing on 4 specific projects. Hopefully, we have time for all of them. And we'll start first with a method I developed to detect life on other planets. Next, we'll talk about an algorithm I developed to predict mineral occurrences on Earth and potentially even on other planets like Mars. Next, we will move on to an ongoing project I have called PI. Yes, PI. I like acronyms when they spell out food. In the project PI, we are trying to find patterns in exoplanet data to tease out how these extremely complex celestial body groups and how that information can be used to answer many astronomical or astrobiological questions. Lastly, I'll present to you some very new and unpublished work on how we are making crystallographic data AI ready. So with that somewhat lengthy prelude out of the way, let's talk about the scientific advances that I promised you at the beginning. Starting off with the very first project, our main goal here was to see if we could develop a reliable method to detect biosignatures. That is, to distinguish between biotic samples and abiotic samples. This project involves some amazing colleagues, each with their own expertise and knowledge. This work would not have been possible without these folks, and of course would not have been possible without the agencies that funded our work. So, what is a good biosignature detection method? Well, a good biosignature detection method should meet four main criteria. One, it must be agnostic, so it should be capable of detecting signs of life different from Earth's biology. Second, it should be probabilistic. That is, the model or method developed must be able to quantify the confidence in the detected biosignature. Third, it must be robust. That is, the method should be consistent and accurate at detecting said biosignatures. And of course, last is that it should be flight ready. So the instrument from which the data is collected should be commonly flown on space missions. As I go through this talk, you will see how we address each of these 4 points with our method. To start off, we used pyrolysis gas chromatography coupled with mass spectrometry to analyze about 134 samples in our initial run. Pyrolysis GCMS involves heating the samples to break it down into smaller components. Next, we use gas chromatography to separate out these components. And lastly, we use mass spec to detect specific molecules from the spectra. We analyzed a good mix of biotic and abiotic samples, including leaves, fossils, meteorites, nylon, etc. Data produced by the pyrolysis GCMS looks like this. It has 3 main attributes. We have rows represented by scan numbers, which is a proxy for retention time. We have columns representing mass to charge ratios. And then we have intensity values for each row and column combination. Most of the times, researchers using PyGCMS identify certain peaks in the data and then use them to identify specific molecules. These identified molecules are then used to ascertain the nature of the sample. We, on the other hand, look at the entire data landscape. We include the positions of the peaks, we include the intensity values, we include the relationships between the peaks. So in this figure, you can see the differences in the aggregated GCMS data for bacteria, plants, petroleum, and meteorite. By looking at the entire data landscape, our model is able to find recurring patterns in the data and uses them to predict the biogenicity of a sample. What you see on this slide is an ultra-simplified workflow of our method. First, we ran 134 samples on our PyGCMS. Next, we pre-processed the data to get it ready for our machine learning models. Once the pre-processing was done, we could get to the crux of our approach, which was building and testing many different classifiers that could predict the abiotic or biotic nature of a sample. We tried quite a few classifiers, including support vector machines, XGBoost, and neural networks. But we found that random forest was the model that performed best. For those who don't know, random forest is a very popular nonlinear model that aggregates over many decision trees in order to improve its predictive capabilities. Once we had our base random forest model, we parallelly improved it and tested its generalizability using feature selection and nested cross-validation. Our final random forest model for the first iteration of our paper was able to distinguish abiotic and biotic samples with close to 90% accuracy. Now that we had our models and predictions, next we wanted to explore and interpret our results. What you see on the screen is a very simple PCA plot showing how similar or dissimilar the analyzed samples are. The first 2 principal components account for about 55% of the variance in the data. The other 18 plus components that we had feature selected contribute still to 45% of the variance of the data, showing you how complex our data truly is. We colored these samples by their biotic and abiotic nature. Well, at least we did so initially, until we noticed that there was a clear bifurcation in the biotic samples spreading to the right. As you can see, The abiotic samples are clumped together on the bottom left in the orange, but the biotic samples start there and spread to the right in two different directions. We examined this trend and found that these two groups corresponded to fresh biotic samples and ancient degraded biotic samples, something that we didn't train the model to identify. It was just able to figure out those differences. That's pretty amazing. Another point I'd like to mention is about why our method is agnostic. So let's say you have 3 samples to test using our method. You can see based on the molecular distributions produced by the sample that they'll assume their position somewhere in the PCA plot. So in my example, you can see that unknown samples 1 and 3 squarely place themselves among existing samples. By the looks of it, sample 3 will probably be predicted as a fresh biotic sample, and that'll be right. But what about sample 2? Sample 2 behaves unlike any of the biotic or abiotic samples that we've trained on. In this case, it could mean one of two things. It could mean, one, that our training set can and should be expanded, which we are already in the process of doing. I'll talk about that a little bit in the coming slides. But the second is a more exciting possibility. The second possibility shows that this unknown sample 2 is unlike any of the biotic or abiotic samples in our training data. So we've identified an anomalous sample for further study, and there's potential for us to find life on other planets different from what we know on Earth. So, to summarize, we've developed a potentially agnostic method to distinguish biotic and abiotic samples. This distinction is not based on the identities of any molecules, but rather on the molecular distributions, which may point to a fundamental difference between abiotic organic mixtures and biotic molecular suits. Lastly, our method is extendable, so it can be used to analyze many different samples, including those from PyGCMS instruments that are used on board existing space missions. I've already talked about why our method is agnostic. So to summarize the other key points for a strong biosignature detection method, a machine learning method like random forest can certainly provide a quantified confidence value in its predictions made, thus making our method probabilistic. With our steps to assess generalizability using nested cross-validation and our resultant model still maintaining close to 90% accuracy, I'm going to consider our method robust at detecting biosignatures. And as I've mentioned, this method is flight ready. The pyrolysis GCMS is one of the most commonly flown instruments in space, so this method can easily be extended to flight-ready missions. So, what's next for us? What do we have planned for this project? Well, there are two big directions we're pursuing for this project: an astrobiological path forward and a paleobiological one. On the astrobiological side, we recently received a $5 million grant from the NASA ICAR program to develop a multi-messenger machine learning model to identify biosignatures. This means we'll be expanding beyond the PyGCMS to include Raman, isotope ratio mass spec, and other instruments. On the paleobiological side, we've so far run 408 samples through our PyGCMS machine, and we've developed a more granular classifier to not just detect the differences between biogenic and abiogenic samples, but also detect the differences between ancient life and modern life, and photosynthetic and non-photosynthetic samples. Look out for our paper that will be published in PNAS very soon. It got accepted last month and it's currently in press. I'll give you one major result as a preview for this upcoming paper. We were able to identify the oldest known molecular biosignature in history from a sample aged approximately 3.51 billion years. With that very exciting result, let's abruptly move on to our next project. We'll be looking at something completely different, but just as exciting: minerals. In this project, I'll talk about how we were able to predict previously unknown mineral occurrences on Earth. The method behind this advance is association analysis, which is built on a data mining algorithm that uncovers association rules based on frequently occurring patterns in the data. It has been used as part of many recommender systems in retail and even in clinical diagnosis. In this talk, I'll tell you about how we used such a method to aid us in mineral exploration. In addition to minerals, we're also planning to use this method to better understand microbial occurrences, and we've already applied it to understand exoplanet occurrences. The idea around mineral association analysis started around 8 years ago when we were discussing the big questions of mineralogy that still remain unanswered. I realized at the time that one of the simplest but most important questions one could ask was where to go find minerals. So this got me thinking about mineral occurrences and how one can use patterns in the known occurrences of these minerals to predict where to find similar currently unknown mineral occurrences. The mineral association analysis method acts like a recommender system where it looks at mineral occurrences the same way Amazon looks at what you've purchased or Netflix looks at the movies that you've watched, and then tries to predict what you may want to buy or watch next. Since this has been a long-term project, there are many more colleagues to highlight here. Specifically, I'd like to call out Shauna Morrison and Jolyon Ralph. Shauna is the lead mineralogist and co-lead for this project, and without Jolyon and Mindad.org, we could not have done this work. The first step in our method is to apply association rule learning to mineral assemblages. We then end up with rules like the ones you see on the screen. Each rule has a support, lift, and confidence value attached to it. These metrics are used to rank the rules. So the first rule, for example, says that if calcite and pumpellyite are found in a certain locality, you're highly likely to find quartz at that locality. The rules I just showed you are the backbone of the predictions that we make. Now, what kind of predictions can we make using these rules? The first is fairly easy to guess, and you already see it on the screen. For any given mineral, we can predict previously unknown localities where that mineral can be found. One such prediction that we made is that you're likely to find tephrite at 4 localities on Earth. A couple of localities in Kazakhstan, one locality in Virginia in the US, and another locality in Sweden. Another example, and this one is straight from our paper, are the localities where you can find critical minerals in the US. Here we are looking specifically at predicted localities for monazite, allanite, and spodumene. These minerals are extremely important sources of lithium, cerium, and other rare earth elements, which we all know are used in some of the major technological advances in modern society. The more astute among you might have already also noticed that we have 2 types of icons in this figure. One is a blue icon and the other is a pink Mindad.org icon. The pink icon shows locality predictions that we've made that have already been ground truthed on Mindad.org. So the predictions that we've made are actually coming true. That's really cool. The second type of prediction we can make using mineral association analysis is to predict the mineral inventory for any given locality. So this is sort of the reverse of the previous examples I've just shown you. For this type of prediction, let's look at Tacopa Basin in California. As many of you may already know, Tacopa Basin is a Mars analog site in the Mojave Desert, and it has been the focus of extensive chemical, mineralogical, and geological studies. It was one of the locations where the NASA Mars 2020 rover was payload tested and ground truthed. Our predictions here show that there's a moderate to high likelihood that these 9 minerals on the table can be found in Tacopa Basin, in addition to the minerals already discovered there. This also brings up the question and possibility that these same 9 predicted minerals may be found in a similar Paleolake environment on Mars. Now, this is an exciting question on many levels, given that there are phases of these minerals that may be of astrobiological interest, because minerals like hematite, pyrite, and gypsum can form through biomediated processes. So I'll wrap this section up with a summary of things that we can learn from mineral association analysis. First, we can search for key mineral resources and predict mineral occurrences on Earth and other planetary bodies. Second, we have predicted the mineral inventory for important localities on Earth and potentially beyond. The third takeaway that I didn't really have time to discuss in this talk was for our exploration of mineralization and mineral occurrences through deep time. If you want to learn more about this type of work, if you want to learn more about the algorithm, the results, the implications, or anything else, please don't hesitate to contact me. I can send you our manuscript, which has been published in PNAS Nexus. The third topic that I want to talk about today is our exploration of exoplanets. This project is called PI, or Planetary Informatics Exploration. The conceptualization for this project started about a few years ago. Our goal here is to study exoplanets using data-driven approaches. We want to see if we can learn more about the characteristics and diversity of these exoplanets. We plan to apply a set of descriptive, predictive, and prescriptive analytics methods to exoplanet data and see if we can form a Hertzsprung-Russell analog for exoplanets. This project started in 2022, and we've seen some excellent work done from my students, mostly undergraduate. So this is a very early career-driven project overall. For this exploration, we decided to start with the NASA Exoplanet Archive, which has about 6,000 odd exoplanets so far and many attributes. To start off our very first study, we decided to only analyze hot Jupiters, since they're the most plentiful in our dataset and they're the easiest planets to observe. To start off on our exoplanet exploration, we decided to go with unsupervised methods like a clustering algorithm to better understand our data. So the first method we used was hierarchical clustering, which is a bottom-up clustering method that starts with creating a distance matrix and then combining the nearest data points to highlight hierarchical relationships in the data. Next, we tried k-means clustering, which everyone here already knows, where in k-means clustering, we group data points based on the geometric centers of each cluster. And lastly, we ran a density-based clustering method called HDB scan, which focuses on grouping closely packed data points together. Next on our list was to choose the optimum number of clusters for our algorithm. Now, there are many different ways and metrics to go about this. My PhD student used this particular method from the reval function in Python. Here, we test the stability of different cluster configurations by training an independent classifier to predict assigned clusters. To evaluate the stability of cluster configurations, we used 4 classifiers, including logistic regression, random forest, the k nearest neighbors algorithm, and support vector machines. The first set of attributes used in our clustering algorithms were planet mass, radius, temperature, and orbital period. After running a comprehensive grid search, we found the top 3 model combinations based on our test error metric. Hyperparameter tuning was then performed on the top 3 models to optimize and analyze our results. Here is an example of one of the model combos and how the results look. Another thing to mention here is that the lower the stability score, the more stable the cluster configuration. So in an ideal world, we're looking for a stability score of 0. And as we can see from this graph, 5 clusters and 7 clusters are the most stable cluster configurations. So we looked at how these clusters separated in the PCA space. There isn't much here. So the more interesting result that we found was when we compared the clusters to the attributes we used in the algorithm. So when you look at the 7 cluster configuration against the 4 features that we used, you can see some pretty interesting groups start to emerge. First, you see this orange cluster emerge, which shows the smallest planets, and then the brown cluster, which shows the coldest planets that are farthest away from their stars. You can also see how the orange group of planets stands out clearly in the plots in the top right. It's a little harder to see how the distributions are split near the central clumped up groups, but I have some takeaways from here too that my students found. So the gray group consists of the most massive planets, and the purple group consists of the hot planets that are bigger than the average hot Jupiter. To summarize our preliminary but promising results, hierarchical clustering combined with KNN show the most stable cluster configurations. We embraced the multidimensionality of exoplanets and built an unsupervised model that separates out subsets of hot Jupiters and helps us find potential boundaries for the smallest planets and for some of the warmer Jupiters. And as I've mentioned a couple of times, these were some of our initial runs that we've conducted on the hot Jupiter dataset. So next step obviously include adding more attributes from the NASA Exoplanet Archive, including stellar attributes. And we'd also like to move past hot Jupiters to some of the other types of planets. Lastly, I wanted to highlight work done by my summer intern Vasundhara Gatne, who has also been analyzing planetary systems as a whole this time, including stars. And she's been using association analysis to understand exoplanet occurrences. Her paper just got published in the Astronomical Journal last month. So if you're interested in reading about that, let me know. I can send you the paper, or please ask me more questions. Last project now, I promise you. Thanks for sticking with me through this long talk. Now this one is probably the newest project of the talk. It's also unpublished as of now. Our papers are currently in review. In this project, we dig into crystallographic data in the form of CIF files. And I present to you all, for the very first time in a formal talk, I think, an R package that we developed to make crystallographic data AI ready. Before we start off, I'd like to highlight the work done by Don Go, who is the lead developer on this project, and he's a software engineer in my group at Carnegie. Now, I'm sure everyone in the audience already knows what CIF files are, but for those who don't, let me give you a very brief intro. Crystallographic information files, or CIF files, are the standard file formats for crystallographic data. It can be data from any mineral or material. This data format has been enforced as the standard by the International Union of Crystallography since the 1990s. Some of the major crystal structure databases include the Crystallography Open Database, the Inorganic Crystal Structure Database, and the American Mineralogist Crystal Structure Database. Between these three and a couple of other data resources, we have information about every natural or synthetic crystalline substance ever analyzed. So, with this much data out there, you would think crystallography is a pretty data focused field, and there'll be tons of softwares and tools available for large scale analysis, right? Well, that's what I thought as well. So, I started working on a large project idea where we can try to identify motifs in crystalline materials. Of course, what I found was that while there are many softwares and computational tools in crystallography, none of them are suitable for big data analysis. For example, crystal structure visualization softwares do exist, but all of them can consume and analyze one CIF file at a time. One CIF file. Similarly, CIF parsers and validators do exist and they can perform some preliminary geometric analysis. But what good is this when even they work on one CIF file at a time? And even if we try to hack into this with some kind of batch processing pipeline, it's slow, really slow. So I was left with an exciting scientific idea and no way to explore it. I had so much data, but no way to use it. Obviously, when my team was faced with this wall, we developed our own software package and our own solution to analyze CIF files and get them ready for AI models. So, with over a year of work, I present to you all, for the very first time in a formal talk, Crysttract, an open source R package that brings crystallography into the age of big data. Crysttract can do many things. I've listed about 9 main features here, but don't spend too much time reading this list. Instead, let's look at a simplified workflow for Crysttract. You start with a single CIF file or a set of CIF files. We've tested up to 1,000 CIF files at a time in each batch, and we're still continuing to scale up. Our package can extract the metadata and unit cell parameters. We then use that information to generate the full crystal structure, additionally using atomic coordinates and symmetry operations. Next, the package can calculate interatomic distances, identify bonds, and calculate bond angles too. One feature we have in our package that's simply missing in any other package around is the ability for error propagation for both distances and angles. I'll briefly walk you through this workflow using a single CIF file as an example. And of course, while I said that this is the simplified workflow for our package, our real workflow looks a little bit like this. You're not expected to read through all of this. I just wanted to show it off a little. So instead, let's talk about our example for this talk, and we'll use that example to continue walking our way through the complex workflow. It's a fairly simple crystal structure of strontium silicide, an orthorhombic crystal that contains just 2 elements. We first parse our CIF file or set of CIF files and use pattern matching to extract all the metadata seen in the red box. Next, we extract the atomic data and the symmetry operators, all stored in tables nested in the CIF file. You can see what that looks like for strontium silicide with the 3 tables on the screen. We then apply a function to calculate the actual coordinates of each atom in the crystal. With this, we have a full unit cell. This should theoretically be the final step in the extraction, but it doesn't quite work like that. At least it didn't for us. With a single unit cell, we sometimes miss out on catching interactions from molecules on the edges or corners of the cell. So we have to expand the unit cell by 1 unit in every direction to catch this information. If this is too computationally rigorous, we have a parameter in our function that can restrict this expansion. But it's probably a good idea to do this so that you don't miss any bonds that should exist. Here is a snippet of the table and what these expanded coordinates look like. You can see we are surrounding 1 unit cell in all directions, giving us 27 total unit cells. Lastly, we take the original atomic coordinates and then calculate distances between the transformed coordinates. We use a real space metric tensor, the formula that you can see in the red on the bottom of the slide, to calculate our distances based on the data that we've collected. Once we have the distances between all the atoms, our package then uses them to predict which of the atoms are really bonded using 4 different algorithms. We can take these bonds to then find the bond angles, which completes finally the data that we need to collect from our pipeline. Here's what the predicted bonds are for strontium silicide. It's a fairly easy example to highlight, but we have found through our detailed testing that our package works on pretty much any CIF file from the ICSD. So that's about 175,000 crystal structures. I'm pretty happy with that. Here's also the snippet for the bond angles and what they look like. As I mentioned earlier, one of the features that isn't available in any other crystallography software or package is the ability to rigorously propagate experimental uncertainties from the CIF file all the way through any derived geometric variables. So on the screen, you can see in addition to the bond distance and angles that are presented to you, you will also be presented with the error values that are propagated through the formulas that I'd mentioned earlier in the talk. In addition to the main workflow, Crystrak provides a suite of post-processing functions that are essential for high-throughput analysis and large-scale scientific studies. First, our filter by elements function allows for the complete removal of bonds involving specific elements. Second, we allow users to analyze atoms based on specific site symmetry described by their Wyckoff positions. Third, we have a function to identify and remove ghost distances produced by site occupancy disorder. And as the first in a series of functions that we're trying to create, we have one that aids exploratory studies in Crystrak, where we have a function that can calculate weighted average network bond distance. As you can see, we've gotten a lot done in the past year with Crystrak, and we absolutely plan to continue with the development and maintenance of the Crystrak package. But what I'm really excited about is that we're finally at the stage where I can start actually building machine learning methods for my intended study of identifying motifs in all crystalline minerals and materials. Of course, the availability of this package provides the foundation for many other different studies in geology and biology to now incorporate crystal structures. So I look forward to the scientific community taking the resource we've made available and rushing towards a new era of scientific advances. Hopefully through my talk, I've been able to showcase the reach and impact that data science methods have on the scientific community. We're able to distinguish biotic and abiotic samples, predict mineral occurrences, characterize the diversity of exoplanets, and make crystallography data AI ready. As a member of IMG and of the geoinformatics and mathematical geosciences community at large, I will end my talk by saying that we are the people who are leading the next generation of scientific discoveries. and I'm very enthusiastic to see where the next decade brings all of us. I'd like to once again thank you all for this award and this wonderful opportunity to present this talk. I'm so sorry I can't be there. I wish I could, but I hope to see you all again at a future IMG meeting or any of the other conferences that we all attend. Thank you.